Hello everyone, welcome to our first lecture on the reproductive system. So in this lecture we're going to cover our introduction to the reproductive system. So we'll talk about basic functions, we'll talk about organs, then we'll talk a little bit about the process of meiosis and some of the numerical chromosomal abnormalities that can occur when meiosis doesn't occur normally. So in our introduction to the reproductive system here, you have your two basic types of reproductive organs here, which are gonads and accessory organs. So gonads are the reproductive organs that are testes in males or ovaries in females, and they perform two of the important basic functions that they produce gametes and they produce sex hormones. Then you have accessory sex organs, which would be ducts, glands, and external genitalia. The duct system is a series of tubes with an opening to the external environment, sort of forms what we call the reproductive tract. Um, so this tract receives secretions, gametes, and transports gametes. Then you have the accessory glands and organs. Glands and organs in or around the reproductive tract that secrete fluids into the reproductive duct system and into other excretory ducts. You have external genitalia, which are structures surrounding or outside of the reproductive tract that support basic functions of the reproductive system, like the clitoris or breasts in females, the penis and scrotum in males. And all these organs help to sort of perform these basic functions. So, kind of overall, the function of the reproductive system is to ensure the continued existence of the species by producing, storing, nourishing, and transporting gametes. So gametes are haploid sex cells. These are spermatozoa or sperm in males and oocytes or eggs in females. To produce the sex hormones, to produce those gametes, and those gametes are going to be involved in fertilization, which you see in the image over here. Fertilization is the merging of haploid male gametes, which are the sperm, and haploid female gametes, which are egg, to produce a new diploid cell called a zygote, which basically grows into the new individual. It also, this occurs only in the female reproductive tract. And then the final function is support developing zygote and fetus, which also occurs only in the female reproductive tract. Now, the first major concept in the reproductive system is meiosis. So meiosis is important because we need to produce these haploid gametes, right? The sperm cells and the egg cells. Now remember, haploid means half the normal number of chromosomes. So remember, the normal number of chromosomes in most cells is 46. If you're going to do fertilization, if you merge two cells with 46 chromosomes, then you'd end up with a new cell that had 92 chromosomes. So you have to create these cells that have half the normal number so that when they merge together in fertilization, they form a new cell with 46 chromosomes, not something else. And so meiosis is really the specific form of cell division for creating those haploid cells, those cells that have half the normal number of chromosomes. So people typically describe meiosis as a form of cell division used to make haploid gametes from diploid stem cells. So in general, the process of meiosis takes one diploid parent cell through 2 rounds of cell division to produce 4 haploid daughter cells. And meiosis, like I said, has 2 rounds of cell division. The first round is what's called meiosis 1, and it has its own phases, which you'll see here: prophase 1, metaphase 1, anaphase, and telophase 1. And then the second round of cell division is called meiosis 2, and that one will have the same phases, but the second version, so like prophase 2 and metaphase 2, anaphase 2 and telophase 2, even though they don't show it to you here in this image, that happens also in meiosis 2. So let's talk about meiosis one. I'm going to focus only on general features of these two rounds of cell division that you'll see in your notes. So you might see a little bit more information in the picture than you see in your notes. So let's focus on what's in the notes. Okay, so in meiosis one, you have a diploid parent cell that divides into two haploid daughter cells. Okay, so because during this round, the number of chromosomes reduces from 46 to 23, the meiosis 1 is often called the reduction division. This is when the number of chromosomes actually is reduced in meiosis 1, not in meiosis 2. Okay? So then what happens in meiosis 2? Meiosis 2 is similar to mitosis in that basically you're going to take those two haploid daughter cells that you made it at the end of meiosis 1 and you're really just going to create an extra copy of them so that you produce four haploid daughter cells instead of just two haploid daughter cells. And those haploid daughter cells that come out of meiosis 2 will end up either being the spermatozoa or the sperm cells or the oocytes or egg cells. Now, one of the things that's important about meiosis that's kind of different between mitosis and meiosis is during meiosis, genetic variation is introduced into the chromosomes on purpose so that your offspring are not exact genetic copies of you. And this is important because it protects the species from extinction due to disease or significant changes to the environment. So like if you have a population of individuals that are all clones, they're all genetically identical. That means that they have the same genetic strengths and genetic weaknesses, every single individual. So let's say they have a genetic weakness to a particular viral infection. Well, that means that that viral infection will wipe out the entire population all at once. So in meiosis, what you do is you introduce changes into the chromosomes, into the DNA, and you do it on purpose so that individuals are genetically different and so that you don't have that same genetic weakness in every individual in your population. That one viral disease is not going to wipe out everyone, and your species continues. Okay? Now, next I'd like to talk about some things that can go wrong in the process of meiosis. And when things go wrong in the process of meiosis, you can get what are called numerical chromosome abnormalities. So numerical chromosome abnormalities are going to be these errors that occur in meiosis that produce gametes with abnormal numbers of chromosomes. So either missing a chromosome or having an extra chromosome, which is kind of what you're seeing in this example here. So like monosomy means you're like missing one of the chromosomes, and trisomy means you have an extra copy of one of the chromosomes. Now, one of the ways that this happens is what's called a non-disjunction. So remember in meiosis 1, you're supposed to be separating homologous chromosomes. You're supposed to be separating basically like mom's chromosome from dad's chromosome. So let's say this is mom's chromosome number 1, and this is dad's chromosome number 1. In meiosis 1, you're going to separate those two so that mom's chromosome number 1 ends up in this cell and dad's chromosome number 1 ends up in this cell. So that's kind of what you're supposed to do. But sometimes when you're doing that, the microtubules that are kind of pulling these things apart can break, or a chromosome can sort of stay stuck to one of the others. And if that happens, then you end up with kind of a situation like this, where both of the chromosomes end up in one daughter cell and the other daughter cell doesn't get any. So you end up with a daughter cell that has extra chromosomes and a daughter cell that's missing chromosomes. And so then, that can trickle down into the daughter cells you get in the end of meiosis 2, where you end up with these cells that have abnormal numbers of chromosomes. And so if these cells are merged in fertilization, then you're going to end up with a zygote that has an abnormal number of chromosomes, either, more than 46 or less than 46, depending on which of the gametes were used. Okay? So I just wanted to give you guys a couple of examples here of numerical chromosome abnormalities. There are a bunch of these, and I'm just illustrating only a couple of them. So the first one is probably one that everybody has heard of, This is trisomy 21. This is commonly called Down syndrome. So basically what gives rise to the symptoms in Down syndrome is basically those individuals just have an extra copy of chromosome number 21. So instead of having 2 chromosome 21s, they actually have 3. So now what does that do to their total chromosome number? Right? If they have an extra chromosome 21, how many chromosomes in total do these individuals have? Is it 46 like normal? No, it's actually 47 because they have this extra chromosome, 21. And then trisomy tells you that. Trisomy 21 means that you have 3 copies of chromosome 21. Okay? So this doesn't happen just to chromosome 21. This can happen in other chromosomes. And I'm just giving you guys one more example. There are others, but I'm just going to give you guys one more, which is the sex chromosomes. So this is going to happen in the sex chromosomes as well. So if a female inherits an extra X chromosome, this is called triple X syndrome because she has 3 X chromosomes. Okay? So again, how many total chromosomes would this person with triple X syndrome have? Right? Would it be 46 like normal? No, the answer, of course, is it would be 47 because they have this extra chromosome. This can happen to males as well. Um, but before I do that, I'm sorry, I jumped the gun here. The next example is Turner syndrome, which is also abnormal chromosome number in females. But this is one where you would have like one of those gametes that's a monosomy that's missing one of the chromosomes. So in Turner syndrome, you have a female who has just one sex chromosome, just an X. She doesn't have a second X. So it's usually kind of illustrated or written as XO, with the O kind of representing the missing sex chromosome. So how many total chromosomes would this individual have? Would they still have 46 like normal? No, remember, this person would be missing one of their chromosomes. So they would actually have 45 total chromosomes, not 46. Last one is Klein filters. This is where you get the trisomy in the sex chromosome in males. So that typically it's like an extra X in Klinefelter syndrome. So you have an individual that's actually XXY, not XY. So they have this extra sex chromosome. And again, this person would have 47 total chromosomes, not 46. All right, so let's look at some questions. Question 1. During fertilization, the male and female gametes merge to form a blank. All right, so the correct answer to this one is B, right? Diploid zygote. Okay, because the male and female gametes, right? The male gamete is sperm, the female gamete is egg. Those are haploid, right? Those are haploid gametes. And so if you merge 2 haploid cells, you're going to make a diploid cell, right? And that cell just happens to be called a zygote. So next question. An individual with Down syndrome has blank total chromosomes. Okay. So remember that this is a trisomy 21, meaning they have an extra copy of chromosome 21, and that will mean, the normal number is 46. 46 plus the extra chromosome means they have 47 total chromosomes.